So this is a cube. Now we're going to move on to real biology. This is a cube that's been haunting my life since Rubicel and Gattuso published it in 2015. I call it the Rubicel and Gattuso Cube of Climate Change Doom, which is basically, this is what we know. We know a lot how single species react to single drivers over short periods of time in response to climate change. And there are a lot of single species study with single drivers, like an increase in temperature and an increase in CO2 over short periods of time, as short as possible. Usually within a generation for multicellular things, or over a few generations for unicellular things. But what we ultimately want to know is what's going to happen over decades or centuries to entire ecosystems that are dealing with real climate change or real environmental change, where multiple things are going to change at once. And so not only are multiple drivers like CO2 levels, and temperature and salinity and light levels all going to change for populations, but also the other organisms there are going to change. So this multiple driver question is not trivial. So I'm going to start actually on this timeline, going from acclimated to adapted. Right? So the first thing you do when you're given a cube like this is you just pick an axis and ignore the other ones. So I'm going to do exactly 2 slides of vocabulary. This is slide 1 of vocabulary. Plasticity. So short-term responses are what evolutionary biologists call plasticity. Plasticity is a change in phenotype that does not require any underlying change in genotype. When you sweat or you shiver, this is a plastic response. This is an extremely awesome, I was going to say extreme plastic response. Plastic responses tend to be reversible. They can be adaptive, but they can also be maladaptive. For it to be a plastic response, all that is required is that it doesn't require genetic change and it happens in response to an environmental cue. It can be catastrophically wrong in terms of survival, but it's still a plastic response. Evolution, on the other hand, is change in the genetic composition of a population over many generations. That's the definition of evolution, right? It can be adaptive, but again, it can be non-adaptive. There are many reasons evolution can be non-adaptive. Evolution is adaptive when natural selection can act. Natural selection can't always act, and I'll get into that. So one of the things we spent 6 or 7 years doing in my lab was saying, okay, well, how do we link plasticity and evolution? And rather than walking you through that, I'm going to give you the punchline, because I feel like I've been talking about it for 6 or 7 years now, which is that plasticity speeds up evolution. All else being equal, genotypes that are more plastic in response to an environmental change adapt more over a longer period of time to that environmental change. And that was very satisfying. I always find it satisfying when you get a clear result and you can write it in 1 sentence, especially if you can write it in red. So I'm going to revisit that experiment because it gave us that clear response, but it produced a number of perplexing responses. So I'm going to walk you through the perplexing responses. This is the experiment we did. This is Elisa Schaum. We didn't do the experiment on her, although it was her PhD project. So she's just gotten an assistant professorship in Hamburg this week, so I'm very pleased. And what we did is we selected Ostriococcus tori, which is a single-celled, it's a pico-eukaryote, and we selected it at high CO2, which is this purple line. At ambient CO2, this was our control. At high fluctuating CO2, and at slightly above ambient fluctuating CO2. And what we predicted was that at high CO2, we select for growth at high CO2. Here we select for plasticity plus growth at high CO2. In the control, this should be selection for plasticity only, and this is a true control. And what we wanted to know, we had 16 different genotypes of Ostriococcus taken from 16 different places on the planet. And what we wanted to know was the genotypes that were more plastic at the beginning of the experiment evolve more in one or any of these treatments than the ones that were less plastic at the beginning of the experiment. The answer is yes. So what I'm showing you here is the ancestral plastic response. So that's a change, in this case, in photosynthesis rate. You can't use change in growth to predict changes in growth. You need to use a trait that's correlated with fitness, but that is not fitness. So this is the ancestral plastic response going from 0 to big. It goes from 0 to 2. So this guy would double their photosynthetic rate when we doubled the CO2. Sorry for cutting off that axis. And this is a direct response to selection. This is 0. So any value that's not 0 is a lot of evolution. And what you can see is the more plastic the ancestor was, the more evolution there was. What you can also see is that slope is negative, which angered me. And poor Lisa was made to measure all of these growth rates many times. So these error bars are very small. And what happened was these ones that evolved a lot not by increasing their growth rate, but by decreasing it relative to the control of the same genotype, which was a little bit shocking to us, right? So we had selected these things for rapid growth, and in 400 generations, which is very strong selection, we had seen a 60% decrease in growth rates under selection for rapid growth in an environment with more food, right? So that was really perplexing, but it was also incredibly consistent. All right, these are 16 genotypes. So we said, okay, maybe we're measuring the wrong fitness proxy. Other people use competitive fitness. Let's compete these guys against a green fluorescent protein infused osteococcus, which is really good fun because then you have green and super green, right? So here we have the growth rates in pure culture from small to big. So the fast growers are on the right-hand panel. And here we have their competitive ability. And what you can see is the faster you grow, the less well you compete in 3 out of 4 cases. So actually the slow growers, when they're put in competition with someone, can ramp it up, which is kind of interesting. These guys have the opposite correlation. These growth rates are extremely high for osteococcus. So our explanation is, in this case, they're growing at almost 1 cell division per day, which is insane for osteococcus. And while they do still outcompete the GFP, they're not actually getting the increase in competitive ability you would expect given their growth rate. So we have three cases of negative correlations between growth rate and your ability to outcompete someone, and 1 case of a positive slope that's way less positive than we think it should be. And these are just the different environments. So this is the control environment. This is the control environment where we selected for plasticity alone. This is the high CO2 environment. And this is a high CO2 environment where we selected also for plasticity. So you can see that it's not about CO2. It seems to be a general thing that within a reasonable range of growth rates, if you grow faster, you're actually a crap competitor. All the competitors do better than GFP because osteococcus hates